68 million years ago, in what's now Montana, a Tyrannosaurus rex moves through a floodplain forest. Muscle, blood, bone. Alive in every sense the word implies. She's pregnant, carrying the next generation inside her. Within days, maybe weeks, she'll be dead. And by every rule biology understands, everything soft about her, every blood vessel, every living cell, should be gone within a few thousand years at most. That's not speculation. That's the settled physics of decay. Picture a bone that is not supposed to exist. Not the fossil, the bone underneath the fossil. Soft, stretchy, wet enough to bend without snapping in half. That's not from something recently dead, that's from a dinosaur that died 68 million years ago. In 2003, a fossil hunting team led by paleontologist Mary Schweitzer excavated a Tyrannosaurus rex out of the Hell Creek Formation in Montana. The specimen was massive, remote, and nowhere near a road. It was too big and too heavy to fly out in one piece, so the crew did what field paleontologists sometimes have to do when a fossil won't cooperate with logistics. They broke the leg bone in half so it would fit inside the helicopter. That crack should have been meaningless. Instead, it opened a window nobody expected. Inside that break, Schweitzer found something that should have crumbled into stone millions of years earlier. Something soft. Something that stretched instead of shattering. Something that looked, under a microscope, disturbingly alive. By every rule fossilization is supposed to follow, that's impossible. Soft tissue doesn't survive tens of millions of years. It's not supposed to survive tens of thousands. And yet, 20 years later, scientists are still finding it, most recently this year, inside the bones of a completely different dinosaur, using tools Schweitzer never had access to. This is the story of how one broken bone turned into a 2-decade fight over whether dinosaurs left behind more of themselves than anyone thought possible. A fight with real skeptics, real counter-evidence and real reversals, and why, in 2026, it just tipped further in one direction than it's ever tipped before. Let's go back to that helicopter. Mary Schweitzer was working with legendary paleontologist Jack Horner's team when they recovered a T-Rex specimen later catalogued as MOR 1125, nicknamed B-Rex. The bone Schweitzer examined afterward wasn't just fossilized rock in the shape of a leg. Under the microscope, she found translucent, flexible structures that stretched like rubber instead of shattering like stone. Branching networks that looked exactly like blood vessels. Round, dark structures inside them that resembled blood cells. Even what appeared to be intact bone-building cells, still holding their original three-dimensional shape. And this wasn't the only strange thing this particular skeleton had to say. Schweitzer would later identify a type of tissue called medullary bone inside it. A reproductive tissue only found in female birds and reptiles actively laying eggs. It meant this specific T-Rex wasn't just a well-preserved specimen. It was a pregnant female, 68 million years after the fact, still carrying evidence of the last thing her body was doing before she died. In March 2005, Schweitzer and her team published their headline findings in the journal Science. Under a sentence that read almost like a dare, soft tissues are preserved within Hindolym elements of Tyrannosaurus rex. According to the textbook version of fossilization, none of that should have been there. Bone is supposed to be a one-way process. Organic material breaks down, minerals seep in, and after enough time, all that's left is rock in the shape of what used to be alive. Soft, pliable, blood vessel-shaped material has no business surviving a fraction of that timeline, let alone 68 million years of it. Schweitzer knew exactly how that would land. Paleontology had spent 300 years building a working theory of fossilization on the assumption that this kind of thing was flatly impossible. She was about to ask the entire field to reconsider that assumption because of what she saw through one microscope. And she was right to expect a fight. 2 years later, in 2007, Schweitzer's team went further. Working with mass spectrometry experts at Harvard, they extracted and sequenced fragments of collagen, the structural protein that makes up bone and connective tissue, directly from the T-Rex sample. 7 protein fragments came back close enough to modern animals to compare. The closest match wasn't a lizard or a crocodile, it was a chicken, lending support to an idea already gaining traction elsewhere, that birds are the living descendants of dinosaurs. That should have been the moment the story became simply amazing dinosaur discovery. Instead, it became a fight. In 2008, a research team led by Thomas Kay published a paper with a much less flattering explanation. Using scanning electron microscopy, they argued that what Schweitzer's team found wasn't soft dinosaur tissue at all, it was bacterial biofilm. Slime, microorganisms that had colonized the porous bone long after the animal died, leaving behind structures that happened to look like blood vessels and cells if you weren't looking closely enough. It's not a crazy idea. Biofilms are everywhere in nature, and they really can mimic biological structures under a microscope. If Kay's team was right, one of the most sensational dinosaur discoveries of the 21st century was actually just 21st century pond scum that had moved into a dead animal's leftover bone. Schweitzer didn't back down. She called the new critique unconvincing, and over the next several years, the scientific back and forth over one T-Rex femur became one of the more entertaining feuds in modern paleontology. Here's where it gets interesting. The people trying to prove Schweitzer wrong ended up building the case that proved her right. In 2009, an independent group reanalyzed the original 2007 mass spectrometry data, the same data Skeptics said was too noisy to trust, and confirmed the protein signals held up. In 2011, Schweitzer teamed up with biochemist James San Antonio to take the fight directly at the biofilm theory. They mapped the recovered collagen peptide sequences onto the known molecular structure of collagen fibrils, essentially checking whether the fragments fit into the specific three-dimensional slots where real collagen sits inside bone. They did. Bacterial biofilm has no reason to replicate that structure. Slime doesn't know what collagen fibrils look like from the inside. Then, in 2016, a separate research team ran the most direct test yet. They grew actual bacterial biofilms on modern, stripped-down bone in the lab and compared the shapes to the structures recovered from dinosaur fossils. The two didn't match, not in shape, not in texture, not in how they formed. If the fossil material really was just slime, it was behaving nothing like slime does when you grow it on purpose. And there was still the biggest question hanging over all of it. Even if this really was original dinosaur tissue, how? What could possibly let a protein survive being buried for tens of millions of years? Around 2013, Schweitzer's team proposed an answer: iron. Blood is full of it in hemoglobin, and iron happens to be extremely good at triggering something called a Fenton reaction, a chemical process that can cross-link proteins together into a much more stable, damage-resistant structure. Almost like a natural embalming fluid the animal was carrying in its own bloodstream the entire time. It's not proof by itself, but it's a plausible mechanism, and plausible mechanism is exactly what a story like this needs to stop looking like a fluke. By the mid-2010s, the T-Rex femur wasn't an isolated case anymore. In 2017, Schweitzer's team went back to a different specimen, an 80-million-year-old duck-billed dinosaur called Brachylophosaurus, and redid the entire collagen extraction from scratch, specifically designed to rule out any possible contamination. The result confirmed: original collagen, 80 million years old. The same wave of research pointed to something even stranger sitting in another team's data: hints of protein surviving in a fossil dated to 195 million years. If that holds up, it would push the boundary of protein preservation back roughly 3 times further than anyone thought was survivable. Each new specimen made the "it's just biofilm, every time, everywhere" explanation harder to hold on to. Biofilms would have to be an extraordinarily consistent contaminant, replicating the same detailed structures, in the same locations, across multiple different dinosaurs, on multiple continents, for the Skeptics version of the story to keep working. Which brings us to this year. A team, led in part by researchers at the University of Liverpool, examined a new specimen entirely. A 22-kilogram sacrum, the fused bone at the base of the spine, from an Edmontosaurus, a duck-billed dinosaur pulled from the Hell Creek Formation in South Dakota. Same rock formation that produced Schweitzer's original T-Rex. Same rough age, roughly 66 million years old, right at the edge of the extinction event that ended the Cretaceous. Their study, published in the journal Analytical Chemistry, stacked 3 independent lines of evidence on top of each other. First, they used a specialized microscopy technique that detects a specific optical property called birefringence, and the pattern they saw was consistent with intact collagen fiber structure, not random mineral buildup. Second, they ran liquid chromatography mass spectrometry, and, for the first time in this specific specimen, precisely quantified hydroxyproline, an amino acid that's essentially a fingerprint for collagen, since it barely shows up anywhere else in biology. Third, and most convincingly, they sequenced actual collagen peptides from the sample, and those sequences matched peptides already independently recovered from a completely different hadrosaur and from Schweitzer's original T-Rex. That last point matters more than it sounds like it should. If contamination or biofilm were faking these results, there's no reason the fake signal from an Edmontosaurus in South Dakota should happen to match the fake signal from an unrelated T-Rex found decades earlier in Montana. 2 independent contaminations don't normally coordinate their answers. The paper first appeared in early 2025, but it's been resurfacing hard in the science press through the summer of 2026, because every time a new specimen holds up, it makes the entire 20-year-old T-Rex controversy look less like an outlier and more like the first sighting of something real. Before we get carried away, let's be clear about what 20-plus years of collagen evidence does not mean. It does not mean dinosaur DNA. Collagen is a structural protein, not genetic code, and it survives very differently than DNA does. Nobody is cloning a T-Rex out of a fossilized sacrum. If you came here hoping for a real-world Jurassic Park pitch, this isn't it, and no credible researcher on either side of this debate is claiming otherwise. What it does mean is something quieter and arguably more useful. A working method for reading fragments of biology directly off bones that are tens of millions of years old, instead of only reading the shapes carved into them. Collagen sequences can be compared across species the same way DNA sequences can, which is exactly how the original 2007 T-Rex fragments ended up pointing toward chickens, real molecular support for the bird-dinosaur relationship, not just skeletal guesswork. It also reopens a question about the fossil record itself. If original protein can survive this long in the right conditions, every museum drawer full of old, previously written-off dinosaur bones is a candidate for retesting with instruments that didn't exist when most of those bones were pulled out of the ground. Nobody currently knows how many specimens are sitting untested, or what fraction of them might hold their own fragments of hydroxyproline, waiting for the right lab to ask the right question. So go back to that helicopter for a second. In 2003, a research team broke a dinosaur bone in half because it wouldn't fit, and by accident cracked open one of the strangest ongoing arguments in modern science. 23 years later, that argument hasn't been fully settled, but it's no longer one lab, one bone, and one skeptical rebuttal away from disappearing. It's multiple specimens, multiple continents, multiple independent teams, and, as of this year, one more 66-million-year-old sacrum that lined up exactly the way the theory predicted it should. Museums around the world are sitting on tens of thousands of dinosaur bones that have never been tested this way. Nobody knows how many of them are quietly holding on to their own fragments of the animals that made them, waiting, like that T-Rex femur did for 68 million years, for someone to finally look close enough. If you want to know what happens the next time somebody does look, subscribe, because we'll be covering it the moment it breaks.